Yeah.
Hi, this is the Cancer Patient Lab, and I'd like to introduce Dr. Paul Mathew, who's joining us. He's with Tufts University. One of our members, David Plunkett, saw him present some very interesting material in a separate session and thought that he would be a good candidate to speak to our community. As you will see with the slide here, he's focusing on prostate cancer and some novel approaches to make it susceptible to immune treatment. And so that's, I think, a futuristic topic that will be of interest to many people because it opens up a whole new avenue of method of action for treatment for people with prostate cancer. With that, turning it over to you, Dr. Mathew.
Thank you. Thank you for this great opportunity to talk to people who are facing this disease and think about it probably every day of their lives, and worry about it, the future. And my personal relationship with this goes back 25 years when I joined as a junior faculty at MD Anderson in Houston in 2001. My mentor assigned me prostate cancer, amongst the genitourinary cancers. I became very interested in the problem over the years in why the cancer behaved in this very stereotypical fashion for the most part, with the spread to bone. And, obviously metastasis, which is cancer spreading from one side to the other. This is how life is threatened, right? It's rarely because it's growing in one spot, although there are exceptions to that. For example, brain tumors don't metastasize. That's how they become dangerous. They grow in an enclosed space. Whereas cancers like prostate express themselves differently by spreading to distant organs, but they do so in non-random manner. So metastasis is a non-random event, right? It's not like the cancer is growing anywhere it pleases. It can only grow where it has fertile opportunity. And there's something unique about the bone marrow microenvironment that is particularly hospitable to prostate, because in at least 70% of men, bone will be the only site of metastasis. And in 90%, the bone is involved. And of course, there are other sites like lymph nodes, perhaps liver and lung to a lesser extent, certainly almost never the brain. So it's a highly stereotyped pattern. And I became, you could say, obsessed with that in the years I was at MD Anderson. And one of the thoughts I had, you know, with so many patients, hundreds of patients, treating them relatively similar way with hormone therapy and chemotherapy, maybe radioisotopes in those days. More recently, PARP inhibitors, a class of drugs for tumors that are mutant for the BRCA genes and so forth. A relatively limited repertoire of therapy, but none of which, in my estimation, had to do with this behavior that I became obsessed with. So this became almost like a Rosetta Stone. Like if you could decode this mechanism of why this gland that sits below the bladder, relatively small in nature, when it develops cancers at high frequency, obviously the most frequent cancer in among men is prostate cancer, right? It's the most common malignancy. But when it does spread, it spreads to the bone in this way. This is a bone scan showing very high volume bone metastases occupying the majority of the skeleton. And in this patient, you may not actually see anything in the lung, liver, bone if you did a CT scan. So this became the obsession. And the report I can give you after, you could say 16 years of effort at Tufts Medical Center working on this problem singularly, is that we think we have a major explanation, not the only explanation, but a major explanation that this is likely mediated by integrins. And I'll tell you about what integrins are. And I'll tell you why two of them, the alpha V and the alpha 5 of these integrins, collaborate, and why this particular antibody that we developed might be the answer to the problem that you're seeing here. And and uh, and with that, I shall move forward. The Tufts does have a patent on this antibody that we developed for this problem. And I hope to tell you something new about prostate cancer that you might not have heard before. You probably heard a lot of lectures from scientists or physicians who come to your forum. And I'm hoping maybe I'll bring a new perspective, and that this new perspective is actually essential for understanding everything about prostate cancer that is dangerous. All right. So first, I'll introduce you to the normal prostate gland. Imagine, for simplified purposes, that it's like a balloon or a sphere that has an outer layer called the basal layer, and it has an inner layer called the luminal layer. So the basal layer is the one that interacts with the environment. It anchors it to the environment. It mediates the transactions with the environment that keeps things in order. It's that thing that specifies order in the gland, you know. So this interaction with the environment is crucial. And it is also the source, you could say the stem cells of the prostate gland. And many glands are organized like this. The breast is also in this way, and there are many others, bladder and so forth. And so these basal layers harbor these stem cells that they can replicate themselves, but they can also have a secondary pathway to differentiate into these luminal cells that have a specialized function. And in the prostate, that specialized function is secretion of enzymes, and one of which is the PSA, known as a marker for screening and monitoring prostate cancer, but also many other things that go into the principal function of the prostate is to liquefy the semen and contribute to the ejaculatory volume. So that's a highly specialized function, and that's regulated principally by testosterone or the androgens. So the basal layer is not regulated by androgens. In fact, it lacks the androgen receptor, whereas the luminal layer expresses the androgen receptor. All right. So this is like some rudimentary points I wanted to make about the gland. It has two layers, the basal interacting with the environment, that is androgen receptor PSA negative, and then a luminal layer that is specialized as secretory functions. It is androgen receptor and PSA positive. This is a cartoon version. And here's a real version over here on the left, these nice, harmonious, well-behaved glands that are benign. And this brown stain is the one that decorates the basal layer. It's a little thin layer, it's a little bit hard to see. And the larger ones that are plump and well-organized here, these are the luminal layers. And this is the lumen. Well, luminal meaning pointing towards the cavity, the lumen of the gland. And this is where the secretory volume is assembled and passed on into the ejaculatory ducts. So that's your fundamental organization of the prostate gland. Why is that important? It is very important, as I will show you next. So firstly, the basal layer and the luminal layer, we can have markers for them, the CD49F marker. If it's high, that's a basal layer. If it's low, it's a luminal layer. You can actually mark them and study them to see how the gene signatures. Obviously, gene signatures means from DNA to RNA, meaning the message from the genome is passed on to RNA, and then the RNA makes the proteins, and the proteins dictate the behavior. So we can look at the array of signatures, of the expression signatures of RNA, and understand what type of behaviors are going on in there. That's called, transcriptional analysis of gene expression signatures. So here's the array of gene expression signatures for the basal layer, and here's what they are for the luminal layer, and I've told you about what they are already. I want to point out two important differences. One is that the basal layer has this so-called mixed signaling and cell adhesion. Remember what I said about the basal layer adhering to the outside, the matrix? Yeah. So it requires this adhesion sort of program to help it work, amongst others. But I'm just calling these to your attention because they're relevant to our discussion that follows. And when you talk about the luminal layer, the ones that you recognize as androgen receptor signaling, well, of course, that is key. But also you might not know the immune response is actually active in the luminal layer, not so in the basal. So that's the normal gland, okay? But what happens if you take cancers that originate from the prostate and use the same paradigm to separate basal and luminal signatures? And what you find in this is the process of scientists separating them and looking at their signatures and so forth, is that benign normal glands and cancerous glands, when separated by this basal and luminal markers, they look very much identical. I'm going to reproduce this slide in a horizontal fashion here, so you can look here and see benign cancer, benign cancer. Look at the way they're being separated by the basal marker, and look at the gene signatures, essentially overlaid, red and green. They look almost identical, don't they? So if you're a normal gland or a cancer, if you're marked by the basal layer, your gene signature is pretty much the same. So what this, and if you're luminal, the same thing. You're carrying the same set of gene signatures, and you can see how very different basal and luminal are. The red and green simply don't overlap, so they're terribly different. So that's a little secret. I don't know how many people actually know this. Surprisingly few. And what this is essentially saying, put another way, is cancers have a very powerful memory of the gland from which they arose, okay? It's not that cancers are either basal or luminal. They contain admixtures of the two, and varying proportions. And as it turns out, the tumors that have more basal than luminal are the more aggressive ones, all right? And here's the data to show that as you go from a low Gleason grade to a high Gleason grade, there's a progressive enrichment in the basal score. That's within the gland of the prostate gland, and in terms of primary cancers of the gland. And when you go from organ-confined disease to metastatic disease, whether it's hormone refractory disease or hormone-sensitive disease, you see the same enrichment in the basal signature, the CD49 high defined signature. All right, remember I said MYC and adhesion and invasive programs. All right, so this is a large study that was done by UCSF and other investigators, taking over 600 men with metastatic castration-resistant prostate cancer. What is castration-resistant? You're on hormonal therapy to control an advanced cancer. The PSA is rising. The scans are showing progressive disease, even though the testosterone is kept low with treatment. That's castration-resistant. And using this paradigm to separate basal and luminal, you can see that about 55% of patients, that's a good proportion of patients, met the definition of basal enriched prostate cancer, and 45% met the definition of luminal enriched prostate cancer. Mind you, just a reminder, this is patients who have been treated with hormone therapy already. So what happens is when you're treated with hormonal therapy, you can move from a luminal dominant androgen receptor regulated cancer to a more basal androgen receptor low phenotype. And I'll talk more about that so-called plasticity of disease. But here you can see there is a nice segregation of these signaling signatures for the basal enriched to the left and for the luminal enriched to the right lower and top left. And once again, I want to point out to the MYC targets in the basal enriched programs here. I mean, there are others too. Those are relevant, but we'll just focus on the MYC for simplicity. And on the bottom right, we'll focus on the androgen response for the luminal, as well as this interferon gamma and interferon alpha, which are immune signatures. So basal and luminal are very different in men with castration-resistant prostate cancer. All right. So this MYC, this is where the problem is, with castration. MYC is a big problem. So what is MYC? MYC is an oncogene that is important in development, that plays a big role, a majority role in over 70% of human cancers. And for the longest time, it's been very difficult to target MYC. There's been a great deal of interest. It is a transcription factor. A transcription factor is something that goes from the cytoplasm of a cell to the nucleus and influences gene expression programs. These have been very difficult historically to target with therapy, although there's some progress now, but rather limited. But what I want to point out is that MYC is central to the basal phenotype, the aggressive phenotype of prostate cancer, and it has many different functions, including promoting gene instability and acquisition of mutations, impairing DNA repair, causing, interfacing with the cell adhesion program that I mentioned, causing tumor cells to proliferate, supporting these tumor cells with metabolic engines, and evading immunity. So all the bad things that cancers do, MYC has a hand in it, collaborating with other partners, no doubt, but it regulates the multiple hallmarks of aggressive cancer, including prostate. And because we have a short lecture, I want to get right to the point and tell you that we think that integrin mechanosignaling, I'll explain what that is. Integrin mechanosignaling has a lot to do with how MYC works in aggressive prostate, but likely other cancers as well. I'm going to tell you what integrins do, but firstly, here's a cartoon of an integrin, alpha 5, beta 1. These are what we call heterodimers. There are two of them together that form a single unit. It's called alpha 5 beta 1 here. And here's another one with alpha V with its partner. And these two are basically partners in crime, if you will. And they collaborate. There's a strong interplay between these two alpha 5 and alpha V integrins in connecting cancer cells, the basal type cancer cells, to the matrix, meaning the inanimate organic material composed of complex proteins, maybe labeled with carbohydrates and the like. But it connects the cell to that matrix and really forms a linkage to the inner cell skeleton. The cells, believe it or not, have their own skeleton. It's called actin cytoskeleton. And that form allows the cell to be, flexible and strong and pliant, but it also is a harbor for signaling processes. And the stiffer the actin cytoskeleton, the more the signaling of a particular protein called YAP, Y-A-P. It's called a yes-associated protein. So YAP will go to the nucleus once the cell is under tension with the microenvironment, with these integrins mediating this tension. And once it's in there, what it does is it does something called bookmarking of MYC and enabling MYC to work. So YAP enables MYC. Over 80% of MYC-controlled sequences in the genome is enabled and potentiated by this YAP. And YAP is coming from that tension I was referring to. Now, what does that do? Well, if you have the stiffness and YAP's in there and MYC is on, what happens is immunity is shut down. And you get a cold tumor. A cold tumor is a bad thing. It means it's not being recognized by the immune system. It's not been infiltrated by immune cells. They cannot destroy this lethal, enemy, if you will. And it's essentially invisible, all right? But if you block these integrins with the bispecific antibody that combats both integrins at once, you disable this whole tension architecture. YAP no longer is active enough to get into the nucleus. And as a consequence, MYC is shut down. And when MYC is shut down, voila, you get an interferon response that's very robust. It starts to secrete, the tumor cell itself starts to secrete what we call chemokines that attract immune cells in there, including natural killer cells and macrophages, to inflame the tumor. Now the cold, invisible tumor now inflamed and infiltrated with tumor cells, which eliminate the tumor. All right, so that's published data, and which I posted, and that's a citation that I'll share with you. So just a little bit more on integrins. It's a little complicated, but imagine here are your heterodimers, the two things, the alpha and beta parts of the integrin connecting to the matrix. Here's a complex architecture to the actin cytoskeleton, just to give us a little bit more mechanical impression of the complexity of this whole process. But essentially, fibronectin is what we believed had a lot to do with why prostate cancer cells got to the bone, because the integrins on the prostate cancer cell were binding to the fibronectin in the bone marrow. That's how it was colonizing the bone marrow. And force matters in biology. I guess that's another takeaway point. I've been making this point, but I'm going to say it again. Force matters in biology. It might be something new for you to think about. And I think you will hear more and more about this in the near future on a diverse array of elite issues. Most recently, I read a paper on early onset colorectal cancers. Those are going up very rapidly in folks below 40. And it could be that there's a problem in the rectum where maybe, who knows, dietary microplastics accumulating and causing stiffening and so forth. And that's changing everything with that interface between the basal layer and the matrix. So here, what I want to make a point about is, suppose you take a cell from the bone marrow and put it on stiff matrix as opposed to soft matrix, you get completely different fates. If it's on a stiff matrix, it becomes a bone-forming cell. If it's on a soft matrix, it becomes a fat cell. So I've simplified this relatively complex diagram to say something very simple. Stiff means one fate, soft means another fate. So just to simplify, force matters in the cell fate, and it certainly matters in cancer biology, and I think might be very central to the lethal phenotype of prostate cancer. So here's some specific evidence that our bispecific antibody, when treating prostate cancer cells, actually controls that signature that I referred to. Here it is again, that basal signature. You see it here. Remember this diagram, top left? The basal enriched signature is the aggressive treatment-resistant phenotype that eludes immunity. Here, that entire signature is under control. Red bars to the left means downregulation. Green bars to the right means upregulation. So we're downregulating that basal signature with this antibody through the mechanism that I proposed and upregulating the immune response. You can see that signature here. And what that results is, in our tumor models in mice, we are eliminating these very aggressive androgen receptor negative prostate cancer cells in animal models very efficiently across a range of doses in both what we call clonogenic assays and established tumors. So we have rather potent evidence that it's not just in a test tube, it's in animal models as well. And that when we can show that the reason that these tumor cells are being eliminated is because of natural killer or NK cells, which are part of the innate immune system in this mouse model. The reason we can show that is we deplete these natural killer cells, the activity of the antibody disappears. So here's the antibody working very well to eliminate tumors in this mice. All these blue signatures are the tumor cells glowing under the imaging, expressing something called luciferase. But if you and here's the antibody eliminating it. But if you deplete that natural killer cells, then the antibody loses its efficacy. Here's the treated with the antibody, but depleted with natural killer cells. Tumors are still the same as the control. It's in two different models. So it's pretty strong evidence that the bispecific antibody is working through the immune system. And finally, hopefully perhaps the last point of evidence here is that when we take tumors from men who have donated their body tissues to a rapid autopsy program at the University of Washington, Peter Nelson and Cole Morrissey collaboration, you combine it with data sets from other metastatic castration-resistant disease or men who have had prostate cancer of early onset. This is a data set from Germany, men who have had prostate cancer below the age of 50. You can see the very strong correlation of this integrin alpha V with the MYC target, which essentially is the point we're trying to make. MYC is at the heart of this basal aggressive phenotype, and the alpha V integrin is a principal determinant of that MYC signature. And so we come to a summary slide here that really illustrates this paradigm of basal and luminal trajectories of prostate cancer. It's basically a recapitulation of normal development, except in complete disarray, because cancers are cells out of position, essentially. And what happens is that the basal layer is supposed to differentiate into luminal cells. So this is true of the normal cell as well as the cancer cells. So the stem cell, the cancer stem cells that are basal-like will generate this androgen receptor positive PSA response positive cell. But if you treat with hormone therapy, right, intensified hormone therapy, dropping the testosterone and blocking it and so forth, you drive the composite identity of the tumor toward the basal phenotype, right? That's just the plasticity that we're referring to. And so these are very different biologies. And I showed you the gene expression program. This is androgen regulated. But as our research indicates, the basal type is YAP-MYC regulated through that stiffness paradigm, the mechanosignaling paradigm. And our bispecific antibody reverses that in an immunogenic way, meaning it sparks the immune visibility of these basal type tumors and brings in all these immune cells to eliminate them. So this is a sort of a simplified thing, but what I said earlier was that tumors are an admixture of these two types of cells. So you have to think about, combined targeting of luminal directed therapy as well as basal directed therapy. And I think that is the future where transformative advances in this disease may come from. That is our conviction, shall we say. Obviously, it's our conviction. But I do think the field will come around to it eventually. Raghav is my partner in crime. In the laboratory, we've been together over a decade, working with a fleet of other MDs or MD PhDs or PhDs in the laboratory, supported by the National Cancer Institute. We're hoping the NIH will continue to fund our research. Obviously, challenging times for researchers nationwide. And we wouldn't have survived. Very proud to say we would not have gotten this far without the support of amazing patient families and who have lost their loved one in the process, of course, sometimes, unfortunately. And so I'm happy to talk about this work or anything else you'd like to talk about. Thank you for your attention.
Thanks for that overview. I think I could almost follow half of the microbiology. And there's some more sophisticated people in our audience. Let me just ask a couple of questions, and then I'll read some of the questions from the chat, as well as people can use the raise hand feature and ask live. Dr. Mathew, we're patients and caregivers, and so one of our questions is that's very pioneering research that you're doing. When might it be accessible in a clinical trial or other ways for patients? When should we expect this to show up in places where we can take advantage of it?
Well, I mean, I wish I could say it would be tomorrow. We are anxious to get this into the clinic. And the reason we think we can in the first place is we know this is a very good antibody. Whenever you're moving research from an academic setting such as ours into the clinic, you need to know things about pharma development of therapeutics that you perhaps wouldn't have known otherwise. And one of those things is, how do you scale up things? Can you scale something up? Can it be made at volume? And does it have all the characteristics of a good drug? And everything that we've done so far, which I haven't talked about, suggests that indeed this antibody has a future. So that's point number one. Point number two is that we have to convince funding partners, whether venture capital or big pharma or small pharma, to partner with us to develop this, because we as a research university cannot do this on our own. So Raghav Joshi and I have formed a spin-out company in order to enable that progression to first in human trials. We obviously have to get permission from the FDA for an IND, investigate a new drug application, jump through all these hoops that they require to show that this is likely going to be safe, describe the clinical trial. So there's a lot that goes in. It could take, I'm not sure how long, but hopefully not more than a year to get this into the first in human trial. We certainly think the science is very robust. The antibody is very robust. We just need the dollars and the team around us, to get it into trials. So it's a huge priority for us.
Okay. Again, from a patient's point of view, they are, let's say diagnosed, they have a Gleason score, and now they're considering options, radiation, hormone deprivation, maybe this treatment. How would you see it fitting in I don't know, the decision matrix of options, and how would it fit? And in particular, I presume you're acquainted with the work of Bob Gatenby.
No.
Okay, so he's someone we should introduce you to. He
Mhm.
uses language very similar to yours. He uses evolutionary biology
Mhm.
and molecular models and mathematical models and game theory to develop. He's been active with bipolar androgen therapy, for
Okay.
example,
Hmm.
and sort of sort of it's in the same way that you are sort of thinking about the molecular model,
Yeah.
and then you're intervening, and how do you intervene with different
Yeah.
options? So how do you think about that kind of treatment options and maybe even combinations or things of that
Yeah.
sort?
Definitely. Well, certainly, we know, having treated men with prostate cancer for a long period of time, how much the treatment for the disease itself imposes suffering on men. Um, whether it's the primary treatment of the gland and its impact on sexual or urinary function, or the utility of hormones and all the impact it has on libido and skeletal and muscle health and so forth. So the questions we ask ourselves is if our findings are of such primary importance, and we as we believe they are, will they have a future in minimizing the impact of other therapies? Can we treat, for example, high risk formats of the disease that haven't even spread yet with this antibody, with a short course of hormone therapy to enable the immune system to act on the tumor as it is in the gland without necessarily operating on it or radiating it? That is a bit of an imaginative leap. I have, I must concede that. But it is along the lines of what we must think of as a research community to make transformative advances in treatment. Clearly, because we think our antibodies allowing the immune system to see and recognize and destroy these cancer cells, we can certainly combine it with immune checkpoint inhibitors, a very established set of therapies in cancer biology that has not been effective in prostate cancer, probably for the very reasons that I have illuminated in our discourse. So it is, if we are essentially tackling the major obstacle to immunotherapy in prostate cancers, I would propose we are, then we may make a transformational advance in allowing immune checkpoint inhibitors to play a genuine and substantive role in controlling the advanced form of the illness, but possibly even the high-risk localized forms of the treatment to minimize the impact of both local therapy and hormonal therapy. For example, a short course of hormone therapy combined with a bispecific antibody and an immune checkpoint inhibitor. That triple combination might serve to eliminate its dual basal and luminal targeting that is immunogenic, meaning it's going to stimulate the immune system, and then the immune system cells can flood in there, create that activity. Who knows if we will transcend the need for local therapy. We're getting there in bladder cancer, for instance. We're combining antibody drug conjugates and immunotherapy and finding complete response rates in very aggressive organ-confined bladder cancers in over 50% of patients. And we're thinking about sparing the bladder. So it's not an outrageous idea by any means. So I think that is the kind of future we do need to visualize. It does demand that kind of bold thinking. Obviously, the science has to be rigorous enough to get us to that point, but that is the direction in which we must try to navigate.
Hey, Rick Davis has a question, which I have a variant on too, but his question is, in layman language, can you translate this theory to clinical application? Does your antibody address the YAP-MYC pathway, and what is it doing? And I have another related question, I think, which is what's the mechanism, like the mechanics of it? When I hear bispecific, I hear latches onto, PSA, PSMA on one side and latches and then has a payload on the other or grabs a T cell on the other. Can you
Yeah,
talk about sort of the mechanics? I think that's what I'm curious about, and I think what Rick is asking
sure,
as well.
sure. I'm not going to put up any more slides. I'll try and use the words if that's okay, because I'll be hunting for slides and stuff. So just try and use my words. So as I said to you, in terms of the mechanism, what is this YAP mix stuff all about? Let me let me say that the basal layer, the outer layer of the gland, the one that harbors the stem cells, is the one that anchors to the environment. And we need these integrins to do that. Integrins are essentially the connecting, connectors of the environment to the interior. And when these are under tension, the whole phenotype of YAP and MYC is triggered. Okay. When they're soft, remember how I showed you, if it's stiff, you get a bone-forming cell in one context. If it's soft, so it's fate, it determines your fate. Force determines the fate. So these integrins are determining your fate because they're transducing this force, right? And so the force is what triggers YAP. Now, YAP is the central hallmark transcription factor of that force YAP is key to that. It's not the only one, but for simplicity, it is a major one and a deterministic one. Because what it does is goes to the nucleus and turns on the MIC switch to the max. It dials the MIC, the volume up, if you will. Without YAP, the MIC volume is it's not off completely, but it's a low hum. But when I'm trying for the right metaphor. So it turns up the volume on the MIC. And when the MIC is up, the immune system is absolutely shut down. Okay. So this is how a tumor escapes immunity, because MIC's volume is turned up to the max by YAP. YAP is in there because its integrins are signaling through force from the environment. And that is the hallmark of these basal type cells that resist immunity, they resist hormone therapy, they resist chemotherapy, pretty much anything you throw at them, except perhaps our antibody or something like it that attacks the central identity of the basal phenotype. And I would say there are very few therapies out there right now that actually do that. Most of them are targeting the luminal phenotype. And so you asked about bispecific, how does it work? Well, it's an antibody that targets these two integrins simultaneously. It is not trying to recruit a T cell. The bispecific that recruits the T cell engagers is one end of the antibody is connecting to a T cell, the CD3, and the other end of the antibody is connecting, say, a prostate cancer cell, say PSMA. Those are your bispecific T cell engagers for prostate cancer. One of them is a T cell, the other one's a prostate cancer. So we're not doing that. We're just simply going at the heart of the identity of the aggressive disease. And you have to target both those integrins. You cannot target one or the other, our researchers indicated. And you cannot use two separate antibodies because that doesn't work very well either, because they found a way around that too. But when you use it in the bispecific configuration, you target both integrins in one molecule, the equation changes, because this time the integrins are now forced into the cell. I didn't talk about all of this, but it forces their degradation. It takes those integrins and takes them off the cell membrane, sends them into the cytoplasm to something called the lysosome, which is essentially a graveyard. It destroys it, and the enzymes that destroy them. And so the integrins are sort of shut down. And this is what allows that whole force edifice to collapse.
Okay.
Was that understandable?
I think it was to me. It was helpful.
Oh, good. Okay, good.
We'll let Rick be the judge. He has another question here. What's the difference between lumen A and lumen B, and why is there a different response to drugs?
Yeah, that's an important question. So, these further differentiations of luminal phenotypes, one of them is much more responsive to the androgen receptor than the others. I haven't used that nomenclature for our work. We've just simply simplified it as androgen receptor regulated or not. And in the architecture that we use, whereas luminal versus, we use the CD49 marker essentially to define basal versus luminal. I think much more work is needed to be done to harmonize all these nomenclatures. But for simplicity's sake, is something regulated by the androgen receptor or not? And if it is not, it is almost certainly a basal phenotype. And that's the type that I think integrins and YAP and MIC are playing the major role.
Okay, Russ asks a question. Does this relate to the PCS class? Luminal D, LP, BN, BI. Is a higher percentage of HSPC man luminal?
At presentation, I mean, there are various ways to skin a classification cat, if you will. And our work has not addressed these variable nuances across multiple laboratories. And in fact, none of these are being used in the clinic in any substantive way as of this moment, although there is a strong push to do that, including bringing in AI programs to define what this phenotype is, and that's also under investigation. But what I will say is that when most people present with prostate cancer, I would say 99% of prostate cancers at the time of presentation will have a androgen responsive phenotype, meaning you drop the androgens, you will see the PSA fall, you will see the shrinkage in the cancer, but you will not see elimination of the cancer. There will always be a residual phenotype, no matter what description you call it, all these various classifications, right? So they have variable sensitivity to androgens, but no one of them will allow for a curative outcome. You may have long-term control, but it is highly variable. And we've not used any of these classification tools in the clinic to decide whether or not to treat somebody with hormonal therapy based on the probability of response, because pretty much everybody does respond, at least initially. So right now I can say just yesterday, I saw two men, both with very high volume bone metastasis, not different from what I showed you. One of them who's been under years of control after hormonal therapy and chemotherapy, and another who didn't even have a few months of control with treatment and the disease is already progressing. And there isn't any one tool, whether you sequence a DNA mutation or that's clinically available in clinical practice to differentiate these two. We simply try and see what happens. So we take this dynamic approach where we treat patients and then we observe the PSA decline. And if the PSA doesn't decline below a certain level, then we know we're in trouble. So we haven't used any of these tools to define our approach. But what we think is behind all of this is a much simplified idea. We're seeking to simplify and collapse all these complex architectures into this driving phenotype of MYC-regulated basal phenotype that integrins control versus the luminal that androgens control.
Thank you. MJ Kui asks, what is your spin-out company called again?
It's called Jolian Therapeutics, Jolian. It's a C Corp with Delaware.
Thank you. Alan Morris has a series of questions, very intrigued by the connection with bone, and the fact that prostate cancer metastasizes in the bone. Why do you think that prostate cancer is not only bone-centric, but also osteoplastic, not osteolytic? I consider this a medical curiosity.
Yeah.
This fundamentally suggests that osteoblasts are activated more than osteoclasts. Do you agree?
Yeah, certainly. I mean, in the osteoblastic, bone, I didn't get into that short format lecture, but the osteoblasts are actually, I showed that force matters. Remember that force matters slide? That was actually a precursor to an osteoblast placed in a stiff microenvironment becomes an osteoblast. That same precursor put in a soft environment becomes a fat cell and a adipocyte. So in the bone environment, prostate cancer cells secrete things that excite and activate osteoblasts. That's called paraneoplastic effect. And these osteoblasts crosstalk with the prostate cancer cells. They are integral to each other in the way they crosstalk and enable each other's proliferation. So prostate cancer is signaling to the osteoblast, and there's a reciprocity coming back. And what we find is that the same integrins, alpha 5 and alpha V, are playing a crucial role in the stroma, in the osteoblast as well. And we've shown that in the paper, which, of course, hoping you'll be able to read and see it's figure 6, I believe. So we show that this osteoblastic phenotype is also controlled by the dual action of these very same two integrins, and we can shut that phenotype off dramatically by combining these two integrin targeting. So in truth, our therapy is targeting both the tumor cell and its immune evasive property, as well as the enabling osteoblastic response. We see that as a duality of, which is the true identity of the cancer, is this essential crosstalk it has with its environment. We think that is the future of advanced cancer therapeutics, where it is recognized that cancers, as I said to you, are non-random. They're non-random because there's a crosstalk with the environment. At the heart of this lethal phenotypes of many cancers is what is the nature of the crosstalk? What is mediating it? And we think that the crossroads of that are these integrins or similar paradigms to be uncovered for individual cancers down the road. And I think that is actually a huge part of the story. So thank you for that question.
I have a question about the diagnostics that might be associated with what you're doing, so that, a companion diagnostic often can predict people that will respond to this. And it gets at the question also, who is this going to be most useful for? Is this all prostate cancer patients, high Gleason scores, whatever? So can you sort of speak to, is there a possibility for a diagnostic that would be tracking some of these things associated with this treatment? And who will it be ideally suited for?
That's a fantastic question. I think this is something that we we believe we have different levels of ideas about. Firstly, we have studied the distribution, the frequency of distribution of these integrins in primary prostate cancers as published data. We find very high expression frequency of these integrins are turned on in the cancer phenotype and absent in the normal gland. So the normal gland, other than the basal layer, the luminal layer does not express the alpha V integrin at all. But as soon as you start to develop higher grade cancers, it's turned on. And it's at high frequency in metastatic disease. It's in 70% of bone metastases in the tumor cells alone, and in 95% of the bone vasculature in tumor metastases and in the stroma. But it's also in other organ sites. So you could say that the frequency of it is high enough across all men with metastatic disease that one might assume that it's a near universal phenotype if you have osteoblastic bone metastasis, for example. But we also want to show that this integrin-MYC association is a more precise way of annotating a tumor's connection to this phenotype. So that's a secondary layer. So one is the clinical. If you see a clinical evidence of metastatic prostate cancer to bone, almost certainly we think these integrins are implicated. But if you're not sure, you have some intermediate phenotype, for example, we do believe that the integrin-MYC transcriptional signature is not something that's actually being studied so far. There are many companies that will sequence your DNA for you, whether it's Foundation Medicine or Caris or Guardant and so forth, right? That's what doctors are ordering for advanced disease, to look for specific mutations that are targetable. But they do not have anything to do with what we believe is the foundational biology of the disease, which is this integrin-MYC association, right? That's actually not being annotated. So as a companion diagnostic, I do believe this is something that needs to be developed. We are actually talking to Caris about this collaboration, and it's something we want to develop. Of course, they have their own business model and how they want to do things, so we have to, get to that point. But there's a pragmatic way of looking at it, is that we believe it's a near universal phenotype for bone metastatic disease. But in other contexts, and obviously we're interested in other cancers as well, will integrin-MYC associations be the calling card that is specific for this therapeutic? And obviously, when you go to clinical trials, it's important to embed such ideas into those trials so that you can validate them further.
And are there, that Alan Morris has, just on that point of other cancers besides prostate cancers where this might be useful or useful at modeling? He says in fibrosing cancers such as pancreatic and prostate cancer, TGF-beta is considered pivotal. How does this fit into the YAP-MYC pathway?
Super question. Well, again, it's in the manuscript, and the citations are relevant. But TGF-beta is a very important player in suppressing the immune system in cancers, okay? TGF-beta does all sorts of things. There's a long list of things it does to block CD8, the cytotoxic T cells, from coming in. It brings in something called T regulatory cells, the ones that suppress the immune system, the one that won all the Nobel Prizes in medicine last year. So TGF-beta brings them in. They suppress T cell function. They increase something called macrophage polarization from M1 to M2, and so forth and so on. But TGF-beta is sourced mostly from the stroma, not so much from the cancer cells. So your colleague mentioned fibrosing, meaning scarring. That is all about that stiffness that I was referring to. So those tumors that are scarred and have high tension have a lot of TGF-beta. And you know why? Because they require the integrin alpha V on these stromal cells to process TGF-beta from its inactive latent form complexed with a latent peptide. When there's traction on it, it activates the TGF-beta, it releases it from its latent peptide, and it's now activated. So stiffness in the stroma activates TGF-beta, and alpha V is key to that. And what we show in our animal models is that in the tumors themselves, when they implanted into the mice, TGF-beta is strongly downregulated with the bispecific antibody. So when we think about, I didn't mention this because for the sake of simplicity, I talked about MYC alone, but we're actually targeting three major pillars of immunosuppression: MYC, TGF-beta, and a third one called EMT, epithelial mesenchymal transition. These three are strong stem cell immune evasive signatures that are used by a host of malignancies. And again, I think these two integrins have a lot to do with it across a wide variety of cancers, largely because they are recapitulation of what I call embryonic integrins. These integrins are key to embryogenesis. You mentioned somebody with an interest in evolutionary biology. Integrins are ancient things. By the way, the person who discovered integrins and fibronectin, Richard Hines, only passed away just last week. From MIT professor, and obviously a legend, and on whose broad shoulders we all stand. So really a tribute. Thanks to him, we have all of this narrative to talk about. But at any rate, EMT is from Bob Weinberg. That he was the one who discovered that at the Whitehead Institute and taught us about that. But at any rate, these three are major immunosuppressive pathways, have a lot to do with these two integrins in specific. There are many integrins, and there's a lot of skepticism about the integrin field, by the way, I should hasten to add, because people have been studying them for a long time, right? But why hasn't there been advances in the clinic? Why? I would say the following. It's because of the interaction between the integrin alpha 5 and alpha V, and the absolute necessity to target both of them with a very effective modality, such as a bispecific antibody, is key to the failures of the past. If they studied only one integrin targeting and the other one is robustly compensating, you're not going to get anywhere. In fact, you could accelerate the disease by targeting one because the other one rears up and says, none of that's going to happen under my watch, and sort of ramps up and sort of can even increase bone formation. We can see bone formation increasing if we block the alpha V integrin without blocking the alpha 5. It's very interesting. So they really have a robust crosstalk, and all the therapies of the past have targeted only one or the other.
Just a quick question on plasticity. I ran into a company that's building a diagnostic. Apparently, as I understand it, before a cell can metastasize, a cancer cell can metastasize, it gets soft.
Oh, yeah?
And you can measure the softness, like you can poke the cells and measure the softness or firmness of the cells and then tell whether it's about to metastasize, because it's like it has to get out of go through a grate as a soft so it can fit through and metastasize. Have you heard of any of this? The company's name is Artitis.
No, I haven't, but I
Do
do.
I get a receipt or they email?
Sorry.
Yeah, it harkens to a concern that we had, which is cancer cells use integrins to anchor themselves, right? And so they establish themselves, say, in a metastatic niche. We call that the niche, a special sanctuary site where cancer cells can hide and establish their survival and evade immunity. So they're there sitting in dormancy. Say you take out a high-grade Gleason 9 prostate cancer, you've had surgery, your PSA is undetectable, but there's a risk that the cancer cell is out there somewhere, you can't see it, PSA, but it's there in dormancy. So you come in with your integrin antibody and release it from its dormant state. Now it's going to do what? Get back into the circulation and start circulating and causing metastasis? This is the theoretical concern, which is harkens back to softening the cancer cell and allowing it to metastasize, right? So could this be a pro-metastatic treatment rather than an anti-metastatic treatment, paradoxically, because of the softening response that you referred to? But the point we would make is the softening is tied into visibility by the immune system, so that the natural killer cells will say, there you are, and eliminate it. So we don't think this is going to be an issue. At least that's not what the science is pointing to, even as that concern that you raised comes to mind.
Okay, Rick Davis has raised his hand. Rick?
Yeah, thank you very much, Dr. Mathew. I'm not a biologist. I'm not even a scientist, so my level of understanding is not great. And one of the things you've done is really shape my perception of basal and luminal cells. Because I always thought basal cells were relatively good guys and luminal cells were relatively bad guys. But now what I'm hearing today is that no, basal cells are the root of all evil in terms of being non-respondent and a cornerstone of castrate-resistant
Mhm.
prostate cancer. And
Yeah.
I just want to ask you a little more about that. I, I, should science be focusing more on basal cells as being the bad guys and not luminal cells?
Well, I mean, I want to say that yes and no, because I think it's if you target only basal cells, the luminal cells will now drift towards the basal and make up for that gap. Or if you target only luminal, as we already discussed, basal will dominate. So the idea should be, yes, we must focus on basal cells, but we must think about combinations of the basal and luminal phenotypes rather than just one or the other. You understand? So if you think about PSMA now, which is all the rage for Pluvicto and all the bispecifics, or the newer fangled ones like the androgen receptor degraders, or the new antibody drug conjugates, many of them are focusing on luminal markers. Almost none are focusing on basal defined markers. So you're sort of seeing a polarized research, clinical research industry working towards hammering away at that PSA, which is fine because that is a bulk of the tumor and you can get space, right? You can even prolong life. But you're not going to cross at least in my mind, a transformative boundary that is defined by that basal phenotype that resists the immune surveillance. I think if you really want to get a crack at a big time impact on this disease, we've got to combine both ideas in something that's tolerable, minimally toxic, has a minimum impact on patients' quality of life, and a transformative effect on long-term outcome.
And that PSMA, is that created by the luminal cells or the basal cells? I mean, it sits on the outside of the
Yeah,
cell,
yeah, it's
of the
principally
prostate cell.
it's principally a luminal type marker, right? So if you see patients who have very low PSAs with metastatic disease, oftentimes a PSMA scan, which you can image the disease with using the
Right.
is very weak or almost
Yep.
negative. And you almost have to use something called an FDG PET scan, the conventional PET scan for that we use
Right.
cancers to see that aggressive phenotype. That's really what we image as the basal phenotype that is weak in PSMA and very strong in FDG PET. That is the most extreme, almost becoming the neuroendocrine prostate cancer, right? Because the neuroendocrine small cell prostate, which I haven't talked about, those are birthed from the basal phenotype. They come from that basal
Uh,
phenotype. They arise
okay.
from that. They drift off into their own
Okay.
trajectory with
Right.
these neuronal type programs, which are also evading immunity, but ticking over very rapidly and spreading to different sites like the liver and causing lytic rather than osteoblastic bone metastasis and sometimes even brain metastasis and so forth. A very difficult customer. Also very immune evasive. But at the heart of that is a MYC variant, the N-MYC as opposed to C-MYC. So there's some kind of a C to N mix switching that goes on to the neuroendocrine, which makes a lot of sense when in terms of a logical thread of evolution from basal to neuroendocrine small cell.
So I'd like to wrap up the recorded part of this session, but always like to give you an opportunity to give a final parting words of wisdom or a summary, key messages, anything you'd like to leave us with.
Yeah, absolutely. I think the revised way to think about personalized medicine in prostate cancer specifically is sequencing the genome, right? Isn't telling us how the cancer is behaving. It doesn't explain the cancer's behaving, how the cancer is behaving. But by thinking about them in basal and luminal trajectories and understanding what defines basal and the central role of integrins in that, that defines the biology. And what happens is all these mutations, whether it's P53 or P10, the BRCA even, they converge on these integrins in diverse ways to explain the disease phenotype. So we need a new paradigm of thinking of combining basal and luminal targeting for the reasons I've explained, and a new way of thinking about personalized medicine beyond genomics sequencing alone.